If you've ever opened your Y-DNA results on FamilyTreeDNA and immediately felt confused by all the numbers, abbreviations, marker values, haplogroups, mutations, and match lists, you are definitely not alone. Many people purchase a Y-DNA test expecting simple answers, only to discover pages filled with technical terminology that can feel intimidating at first glance. Suddenly, you're looking at things like genetic distance, string markers, SNPs, terminal SNPs, haplogroups, and phylogenetic trees, and it can quickly become overwhelming. The good news is that these concepts are actually far more understandable than they initially appear once they're broken down into plain language. In this presentation, we're going to walk step by step through how Y-DNA testing actually works, why the Y chromosome is so powerful for genealogy, how mutations accumulate over time, how paternal matching functions, and how genealogists interpret these results in real-world research. We'll also discuss how FamilyTreeDNA became the leading company for Y-DNA testing and why their database remains one of the most important resources in genetic genealogy today. By the end of this presentation, those numbers and abbreviations should begin making a lot more sense, and you'll understand why Y-DNA testing has become one of the most powerful tools available for studying paternal ancestry and human migration. Y-DNA refers specifically to the Y chromosome, which is one of the two sex chromosomes in humans. Biological males inherit one X chromosome from their mother and one Y chromosome from their father. Biological females inherit two X chromosomes and therefore do not carry a Y chromosome. What makes the Y chromosome especially important in genealogy is that it passes down the direct paternal line with very little change from generation to generation. Unlike autosomal DNA, which recombines and mixes every generation, the Y chromosome remains relatively stable across centuries. That means a modern man may still carry a remarkably similar Y chromosome to an ancestor who lived hundreds or even thousands of years ago. This creates a direct biological pathway through paternal history from father to son to grandson, then grandson to great-grandson. Because surnames in many cultures also traditionally pass from father to son, Y-DNA became incredibly valuable for surname studies and paternal lineage reconstruction. In many ways, Y-DNA functions almost like a biological time capsule moving forward through generations. Since women do not inherit a Y chromosome, they cannot directly take a Y-DNA test themselves. However, women absolutely can still research paternal lines using Y-DNA by identifying appropriate male relative who carries the Y chromosome from the target paternal line. For example, a father, brother, paternal uncle, or male cousin from the surname line. This is incredibly common in genetic genealogy research. Y-DNA testing becomes especially powerful in situations involving unknown parentage, adoption, surname studies, colonial ancestry, and difficult paternal brick walls. In some cases, Y-DNA can preserve evidence of paternal ancestry that no longer exists in historical records. This is also why testing older generations whenever possible is so important. Once a direct paternal line ends without sons, that particular Y chromosome can disappear permanently from future generations. While many major DNA companies focus primarily on autosomal ethnicity testing, FamilyTreeDNA invested heavily in Y-DNA testing long before most competitors entered the field. Today, FamilyTreeDNA remains the industry leader for advanced paternal lineage testing. Their Y-DNA database is one of the largest in the world and includes surname projects, haplogroup projects, regional studies, clan projects, and advanced SNP analysis. FamilyTreeDNA offers multiple levels of Y-DNA testing. Lower tests, such as Y37 and Y67, provide introductory matching and paternal comparisons. Higher-level tests like Y111 provide much stronger genealogical resolution. Their most advanced test, Big Y700, examines massive portions of the Y chromosome and continues contributing to ongoing scientific discovery by identifying entirely new SNP mutations and expanding the worldwide paternal tree. For serious Y-DNA research, FamilyTreeDNA is generally considered the gold standard platform within genetic genealogy. Although the Y chromosome remains relatively stable, it does still mutate gradually over time. These mutations are incredibly important because they create measurable genetic differences between paternal lines. Imagine a direct paternal line: grandfather, father, son, and grandson. Each generation receives nearly the same Y chromosome as the previous generation. However, occasionally a mutation occurs. That mutation is then passed forward to future descendants of that branch. Over long periods of time, these accumulated mutations create branching paternal lineages that genealogists can compare and analyze. This allows researchers to estimate how closely related two paternal lines may be and approximately how far back their shared paternal ancestor lived. In many ways, Y-DNA behaves almost like a slowly evolving biological surname system. String markers are one of the foundational concepts in Y-DNA testing. String, or STR, stands for short tandem repeat. These are repeating sections of DNA found at specific locations along the Y chromosome. Different men can inherit slightly different repeat counts at these locations, and those differences are what genealogists compare when evaluating paternal relationships. For example, one man may have a marker value of 13 at a particular location, while another may have 14. That single difference represents a mutation event somewhere along the paternal line. FamilyTreeDNA compares dozens or even hundreds of these string markers between testers. The more markers tested, the stronger the comparison, the more accurate the matching, and the better the genealogical resolution. This is why higher-level tests like Y111 are generally much more useful for serious genealogy than lower marker tests. At low marker levels, unrelated paternal lines can sometimes appear deceptively similar. Higher marker counts help separate genuine relationships from coincidental similarities. Genetic distance is one of the most important concepts in Y-DNA interpretation. FamilyTreeDNA compares your string marker values to another tester's string marker values and counts how many mutations separate the two results. That mutation count becomes the genetic distance. For example, if two men match perfectly at all tested markers, their genetic distance is 0. If one marker differs, the genetic distance becomes 1. If 4 markers differ, the genetic distance becomes 4. A simple way I often explain this to people is that, on average, one measurable string mutation tends to occur approximately every 5 to 7 generations. That does not mean mutations occur on a perfectly predictable schedule, because mutation rates vary between families and markers, but it provides a useful genealogical estimate. Generally speaking, smaller genetic distances suggest a more recent shared paternal ancestor, and larger genetic distances suggest the common ancestor lived farther back in time. However, one of the biggest misconceptions in Y-DNA testing is assuming that a low genetic distance automatically proves a recent relationship. DNA inheritance is statistical and probabilistic. Some paternal lines remain genetically stable for many generations. Others accumulate mutations more rapidly. This is why genealogists combine DNA evidence with traditional records, geography, surnames, migration patterns, and documentary research. A mutation simply means that a marker value changed somewhere along the paternal line. For example, a grandfather may carry a marker value of 13. His son inherits 13. Then a grandson inherits 14 due to a mutation event. That new value is now inherited forward through all descendants of that branch. That single measurable change becomes one mutation, one step difference, one unit of genetic distance. Over many generations, additional mutations continue accumulating. Eventually, these mutations create distinct paternal branches that researchers can identify and compare. This gradual accumulation of mutations is one of the reasons why DNA functions almost like a biological timeline. By counting how many mutations separate two paternal lines, genealogists can estimate approximately how recently those lines may have shared a common ancestor. Again, these are statistical estimates, not exact clocks. But the process allows researchers to reconstruct paternal lineage relationships in extraordinary detail. SNP stands for single nucleotide polymorphism. That sounds extremely technical, but in simple terms, an SNP is simply a mutation that occurred at a specific location on the Y chromosome. Unlike STR mutations, which happen more frequently and are useful for recent genealogy, SNP mutations occur much more slowly over long periods of time. When a new SNP mutation occurs, it creates an entirely new branch on the human paternal tree. These major branches are called haplogroups. For example, haplogroup R is one enormous paternal branch. Within R are many smaller branches. Within those branches are even smaller subbranches. Every modern paternal lineage belongs somewhere on this worldwide branching structure. Haplogroups help researchers study ancient migration, population movement, human settlement, and deep ancestral origins. This is why Y-DNA testing is valuable not only for genealogy, but also for anthropology and population genetics. The phylogenetic tree is one of the most fascinating concepts in Y-DNA research. Think of it as a giant branching map of human paternal ancestry built entirely from mutations. Every time a new SNP mutation occurred somewhere in history, it created another branch on the tree. That mutation was then passed forward generation after generation to future descendants. Over thousands of years, these accumulated mutations created the enormous worldwide paternal tree study today. At the very top of the tree are humanity's earliest known paternal lineages. As the tree branches downward, we move into increasingly recent paternal branches and family divisions. This is why haplogroup names become longer and more specific over time. For example, RM269 and RBY1 54784. Each additional mutation creates another subbranch beneath the larger parent branch. Modern testing, especially Big Y700, continues discovering entirely new branches on the phylogenetic tree even today. In many ways, the phylogenetic tree is a living scientific reconstruction of paternal human history. One of the most common points of confusion in Y-DNA testing is the difference between a haplotype and a haplogroup. Although the words sound very similar, they describe completely different things. A haplotype refers to your STR marker signature. In other words, it is the pattern of STR values that genealogists compare when evaluating recent paternal relationships. Your haplotype is what allows FamilyTreeDNA to identify close Y-DNA matches. A haplogroup, on the other hand, refers to your deep ancestral paternal branch defined by SNP mutations. So in simple terms, strings help with recent genealogy, and SNPs help with ancient ancestry. A haplotype may help determine whether two men share a common paternal ancestor within genealogical time. A haplogroup helps identify where a paternal line fits within the much larger story of human migration and ancient population history. Both concepts are extremely important, but they operate on very different timescales. Convergence is one of the most important advanced concepts in Y-DNA interpretation. At lower marker levels, two completely unrelated paternal lines can sometimes appear deceptively similar. This occurs because certain string patterns may resemble each other simply by coincidence. In other cases, some string markers mutate so slowly that unrelated lines can remain broadly similar across limited comparisons. This is why lower-level tests such as Y12 or sometimes even Y37 can occasionally produce misleading impressions of relatedness. Higher marker testing helps reduce this problem significantly. Tests like Y111 and especially Big Y700 provide far greater resolution and help distinguish genuine relationships from coincidental similarities. This is also why experienced genealogists rarely rely on a single low-level match when drawing conclusions. DNA evidence must always be evaluated alongside surnames, geography, historical records, and broader matching patterns. One of the biggest misconceptions in Y-DNA testing is the idea that mutations function like perfectly predictable clocks. They do not. When genealogists discuss mutation rates, we are discussing statistical averages across large populations. For example, the commonly referenced estimate of one measurable string mutation every 5 to 7 generations is only an approximation. Some paternal lines may go many generations without any measurable mutations at all. Other lines may experience multiple mutations in relatively short periods of time. Different string markers also mutate at different rates. Some markers are highly stable. Others mutate more rapidly. This is why Y-DNA interpretation always involves probability rather than certainty. DNA provides ranges, likelihoods, and estimates. Traditional genealogy remains essential for building accurate conclusions. Big Y700 is widely considered the gold standard for advanced Y-DNA testing. Unlike lower marker tests, which examine only a limited number of string markers, Big Y scans massive portions of the Y chromosome itself. This allows researchers to identify new SNP mutations, refined paternal branches, and highly specific lineage divisions. Big Y can sometimes distinguish paternal branches formed only a few hundred years ago. That level of detail is incredibly valuable for surname studies, clan projects, colonial ancestry research, And deep paternal reconstruction, another important feature of Big Y, is that it actively contributes to scientific discovery. As more men test, entirely new SNP branches continue being discovered and added to the worldwide Y-DNA phylogenetic tree. In many ways, Big Y users are helping expand humanity's understanding of paternal history in real time. A terminal SNP refers to the most refined known branch of your direct paternal lineage. Think of it as the furthest known endpoint of your paternal branch on the phylogenetic tree. For example, a broad haplogroup like RM269 may contain thousands upon thousands of smaller descendant branches beneath it. As more SNP mutations accumulated over time, increasingly refined branches formed. A terminal SNP identifies the newest currently known branch that defines your direct paternal line. Some terminal SNPs may represent family divisions that formed thousands of years ago. Others may reflect branching events from only a few hundred years ago. As Big Y testing expands, many family groups continue discovering entirely new terminal SNPs unique to their own paternal branches. When people first begin looking at their Y-DNA matches on FamilyTreeDNA, one of the first things they usually notice is the genetic distance column. At first glance, those numbers may not mean much, but they're actually one of the most important pieces of information in Y-DNA interpretation. FamilyTreeDNA compares your string markers to another tester's string markers and counts how many mutations separate the two results. If two men match exactly at Y111 with a genetic distance of 0, that suggests a very close paternal relationship somewhere within a genealogically meaningful time frame. If the genetic distance increases significantly, the shared paternal ancestor likely lived much farther back in time. However, DNA inheritance is statistical, not perfectly predictable. This is why genealogists combine DNA evidence, documentary evidence, geography, surname patterns, and migration history. The strongest conclusions come when all forms of evidence support each other together. This example demonstrates how genealogists analyze Y-DNA matches in real-world research. In this scenario, both testers share a low genetic distance, the same surname, and similar geographic origins. Individually, none of these factors alone prove a relationship, but together, they begin forming a strong pattern of evidence. Genealogists would then begin comparing family trees, historical records, migration patterns, census records, wills, land records, and documentary timelines. DNA provides biological evidence. Traditional genealogy provides historical context. The strongest research conclusions come when both support each other together. TMRCA stands for time to most recent common ancestor. Using mutation rates and genetic distance, researchers can estimate approximately how recently two paternal lines may have intersected. For example, a genetic distance of 0 at Y111 may suggest a relatively recent shared paternal ancestor. A larger genetic distance may indicate that the common ancestor lived much farther back in time. However, it is extremely important to understand that TMRCA's estimates are statistical probability models. They are not exact historical dates. DNA cannot tell us the exact name of the ancestor, the exact year the ancestor lived, or the exact relationship path. Instead, DNA helps narrow the possible genealogical timeframe. Traditional documentary research is still necessary to identify the actual historical individuals involved. One of the reasons Y-DNA became revolutionary for genealogy is because surnames and Y-DNA often follow the same paternal inheritance pathway. Historically, many cultures pass surnames from father to son. The Y chromosome follows that same inheritance pattern. Because of this, Y-DNA testing allows researchers to determine whether multiple families sharing the same surname are actually biologically related through the paternal line. Before DNA testing existed, genealogists relied entirely on paper trails, oral history, and assumptions. Y-DNA introduced direct biological evidence into surname research for the first time. This transformed genealogy in extraordinary ways. Researchers could suddenly identify unrelated surname groups, hidden paternal branches, migration pathways, ancient clan divisions, and previously unknown lineage connections. Surname projects are one of the strongest and most unique features of FamilyTreeDNA. These collaborative projects allow researchers from around the world to compare Y-DNA results connected to specific surnames, regions, or haplogroups. Project administrators organize results into related family branches and identify patterns within the data. These projects often uncover branch separations, migration routes, unrelated surname lines, ancient paternal origins, and hidden family connections. In many cases, surname projects become long-term collaborative research communities involving historians, genealogists, and DNA researchers working together. For serious paternal lineage research, surname projects can be extraordinarily valuable. This can be one of the most sensitive aspects of Y-DNA research. Historically, surnames and biological inheritance did not always align perfectly. Sometimes Y-DNA reveals that a paternal line does not genetically match the expected paper trail. This may result from adoption, informal surname inheritance, non-paternal events, step-family integration, or historical family circumstances. It is important to remember that DNA does not invalidate family identity or lived family history. Instead, DNA provides another layer of historical evidence. Genealogists must carefully evaluate both documentary evidence and genetic evidence together. These discoveries should always be approached respectfully and thoughtfully. Although Y-DNA is incredibly powerful, it is also highly specialized. Every person has thousands of ancestors. Y-DNA examines only one narrow paternal pathway among all of those ancestral lines. Because of this, Y-DNA cannot represent your entire ancestry, replace traditional genealogy, explain all family relationships, or function as a complete ethnicity analysis instead. Y-DNA works best as one tool within a much larger genealogical research strategy. The strongest results come from combining Y-DNA, autosomal DNA, documentary evidence, historical context, family trees, and migration analysis. DNA alone is rarely the complete answer. But when combined with traditional research, it becomes extraordinarily powerful. One of the most remarkable aspects of Y-DNA testing is its ability to preserve biological history across generations. Records can disappear, names can change, paper trails can end, but the Y chromosome continues moving forward generation after generation. Y-DNA allows researchers to reconstruct paternal lineages, study ancient migrations, confirm surname connections, identify hidden family branches, and connect distant relatives across the world. In many cases, Y-DNA preserves evidence of paternal ancestry that was never recorded historically. That is what makes it such a powerful tool in both genealogy and population history. One of the most powerful realizations in Y-DNA research is understanding that every living paternal line represents an unbroken chain stretching back through history. Wars, migration, settlement, survival, exploration, family formation, all of it exists silently within the Y chromosome. Genealogy stops becoming just names on paper. Instead, it becomes the study of living human continuity across generations. Every paternal lineage that survives today represents countless generations of inheritance stretching back through time. That perspective transforms Y-DNA testing into something far larger than simple matching. It becomes part of the larger story of humanity itself. Y-DNA testing combines science, genealogy, history, and anthropology into one remarkable research tool. Whether someone is exploring a surname mystery, an unknown paternal line, a colonial ancestor, an adoption case, or deep ancient ancestry, Y-DNA can provide answers that traditional records alone often cannot. As testing technology continues advancing, our understanding of paternal lineage history continues expanding as well. And through companies like FamilyTreeDNA, genealogists today have access to tools that previous generations of researchers could only dream about. Thank you so much for joining me for this presentation on Y-DNA testing and paternal lineage research. I hope this helped make some of the concepts behind Y-DNA testing feel less intimidating and more understandable. In future presentations, we'll continue exploring advanced topics in genetic genealogy, including haplogroups, autosomal DNA, chromosome mapping, ethnicity estimates, triangulation, ancient ancestry, surname projects, and real-world DNA case studies. If you enjoyed this presentation, please like, subscribe, and share.